In this video, we're going to talk about heredity and specifically Mendelian heredity, which means we're going to be talking about this guy, Gregor Mendel, and here he is working with his pea plants, and he did a lot of crosses with pea plants. He made pea plants famous. So what he did, he was first off not just a monk, he was a highly educated person. He knew a lot about math, he knew a lot about statistics, and he knew a lot about plant biology. And he knew that plants had male and female parts, and that they typically, in the case of the pea plant, self-pollinated. So what he did is he said, what happens if I take two different varieties like white flowered pea plants and purple flowered pea plants and cross them together? And when he did that, he removed some of the male pollen from the purple and added it to the eggs of the white, and he produced peas that when they grew became all purple flowered. So based on that, he came up with his rule of dominance. Now we say it's a rule of dominance because there are exceptions to Mendel's rule, right? There's incomplete dominance and codominance. We'll talk about those later. But for this, for his work, he basically, when he ever crossed two different varieties or two different traits, he found that one was always dominant. Okay, so purple versus white flowers, he crossed them and he got all purple plants. Had Mendel just stopped with one cross, that would have been it. He would have just come up with his rule of dominance. But he crossed them for several generations. So he looked at these hybrids that were produced, these purple plants produced from the first cross, what we call the F1 generation. He said, what happens if I cross these? And when he crossed these purple plants from the F1 generation, he got an interesting thing in what we call the F2 generation. This just means the second generation. He got about a 3-to-1 ratio of purple plants to white plants. So clearly this was not a blending. At the time, people thought traits were just blended together, just like paints. We mix purple and white, we're going to get sort of a lighter purplish color. And what he saw clearly was that these traits did not blend together, that they remained separate, and that one was usually dominant, and that when he crossed these individuals that were hybrids, he got this 3-to-1 ratio. So he didn't just look at flower color. He did work extensively with pea plants, but he came up with this rule of segregation, that when you have something that is a hybrid, even though it shows the dominant, it will still pass on the recessive trait. And he came up with a description for that he called the rule of segregation, that these alleles segregate, they don't just blend together. So he looked at a lot of different traits. He looked at purple and white. He crossed them again. When he crossed the F1, he all got close to this 3-to-1 ratio in the F2. He looked at axial versus terminal flowers, and again he got in the F2 generation close to a 3-to-1 ratio. Yellow versus green, round versus wrinkled. So this, based on the pea plant, supported this idea that yes, one trait is always dominant, but when I have a hybrid, a heterozygous individual, I'm always going to see this 3-to-1 ratio in the offspring. And we know today, Mendel didn't know anything about chromosomes or genes, but we know today that our genes exist as these pairs on homologous chromosomes. So if you're a hybrid, if you're heterozygous, you will have differing alleles. Allele is a variation. So those individuals, those hybrids that he crossed, had on one chromosome an allele for purple, and on the other chromosome an allele for white. And when they segregated during meiosis, half of the sex cells got the white, half of the sex cells got the purple. So again, to look at this, again, relating meiosis to this idea of heredity, when we have individuals that are hybrids that have differing or heterozygous characteristics, what we'll see is, again, during that meiosis 1, when we separate homologous chromosomes, half of them will get the dominant allele, half will get the recessive allele. And this is important when we look at heredity problems and trying to figure out the probability of traits being passed on. So this is just relating meiosis to this idea of the principle or rule of segregation that Mendel first noted. So a couple terms we need to go over when we talk about heredity. First off, I have to talk about what I see, and the phenotype is what is seen, seen. So what do I see here? I see purple flowers, or over here I see white flowers. That's the phenotype, the observable trait, okay? What you see in the genes, or what you don't see, but what is in the genes, is the genotype. Now remember, since all of our genes exist on homologous chromosomes as allele pairs, we have to have 2 of each of these genotypes, and we'll use symbols for them. So since purple is dominant and white is recessive, we'll say big P is the gene for purple. So if it's a purple flower, it could be big P, or it could be big P, little p. If it's a white flower, we're going to say since that's recessive, little p. We're not going to go with W because we want to show a dominant and recessive nature. So since P is dominant, it gets the big P. Since little p is recessive, for white, it gets the little p. Okay? So Mendel's third rule, now he said, okay, what happens if I look at multiple differing traits? So for example, I know that white peas are dominant to green peas, and I know that round peas are dominant to wrinkled peas. When I cross a yellow round pea with a green wrinkled pea, what will I get? So you might say, okay, well, what's going to happen in the first generation is I'm going to get all yellow round. And that's exactly what you get. That's what Mendel got. Then when he crossed those individuals, again, you might think that you'd get this 3-to-1 ratio of yellow round to green wrinkled, given this idea of dominance and segregation. That's not what he got. And what he got led to his third rule, which is this idea of independent assortment. What he got was this 9-to-3 to 3-to-1 ratio of yellow round, yellow wrinkled, green wrinkled. Well, why is that, and what does this independent assortment mean? Well, again, today we know that these different traits are on different chromosomes. So during meiosis, they all segregate independently. What that means is that they assort independently. So for example, we don't just have the dominant yellow and round staying together, right? We might have the yellow and the round. We might have the yellow and the wrinkled. We might have the green and the round, or we might get the green and the wrinkled. So what we're showing here are all the different possibilities within the sex cells, the sperm and the egg, and when we combine them through fertilization, we have all of these different possibilities. Now in freshman bio, I didn't make you guys do these 2-trait crosses. They're just really complicated and hard and frustrating and easy to make mistakes. But in AP biology, on the AP exam, you do have to know how to calculate these. So now we're going to look at a quick, easier way to solve these what are called dihybrid or 2-trait crosses. So here's our cross, yellow and green peas again. But when we look at that, first off, if we're crossing 2 individuals that are both heterozygous for 2 traits, big R, big Y, little y, crossed with itself, we can really consider that 2 monohybrid crosses. That means that this cross, which makes this really big 16 square, confusing Punnett square where it's easy to make a lot of mistakes, can actually be broken down into 2 4-square Punnett squares. Big R, little r, big R, big Y across big Y, little y. Now we know what these are. This is going to give me a 3-to-1 ratio. This is going to give me a 3-to-1 ratio. So if I look at the simpler Punnett square for big R, little r, I get this 3 big R to 1 little r, 3 round to 1 wrinkled, and the big Y, little y, I get a 3 yellow to 1 green. Okay, I'm just noting here the phenotypes because ultimately that's what I need to figure out. So if I say I've got this ratio within my round and wrinkled and this ratio within my yellow to green, I can simply multiply this times this and I get this. Okay, so now if I say how many round yellow peas will I get out of 16? Well, 3 times 3 is 9 round yellow. Okay, how many round green will I get? Well, 3 times 1 is 3, and you can see I get 3 round green. Okay, how many wrinkled yellow will I get? Again, 3 yellow wrinkled, and then how many wrinkled green will I get? I'll get 1 times 1 is 1 out of 16. So you can use math to more quickly solve these dihybrid problems. And in class, we'll do a lot of practice with these so you get very comfortable and you can very quickly calculate these ratios. But that's it. This is an introduction to Mendelian heredity, Mendel's 3 rules of heredity that we're going to be using. And then we're going to look at some of the exceptions to Mendel's rules, which make heredity a little bit more tricky, But once you understand this foundation, it's really easy to see how these exceptions play in. So that's it for this video.